Welcome you all, myself S.S. Mahamuni, Assistant Professor from the Department of Pharmaceutical Chemistry, Satara College of Pharmacy, Satara, affiliated to Dr. Babasaheb Ambedkar Technology University, Luneri. Today we are going to see the practical of the subject Instrumental Method of Analysis from the final year B Pharm. The number is 10th, that is circular paper chromatography. So we will start with the first, that is the aim. So aim of the today's practical is to separate and identify of given mixture of amino acid by circular paper chromatography. So this is of the method, that is the circular paper chromatography. We want to know about how to do and how to perform this circular paper chromatography. Then we will move towards the objective. So the objective of this experiment is to study the experimental techniques of paper chromatography and to develop the paper chromatogram of amino acid. Then to study the circular paper chromatography, to study the how to determine the RF value and how to identify the separated and analytes by comparing the RF value with standard, and to study the qualitative aspect of the circular paper chromatography. Next is the principle. So it is the paper chromatographic technique. So this technique is a type of the partition chromatography in which the substances are distributed between two liquids. One is usually water, which is held in a fiber of the paper called as the stationary phase, and other is a moving liquid or the developing solvent called as a moving phase. So both of the phases, that is the stationary phase and mobile phase, are liquid in nature, so that the partition phenomena is playing important role for the separation of the component. So the component of the mixture to be separated migrate at a different rate and appear as a spot at a different part of the paper according to their partition coefficient. In this technique, the test solution is applied as a small spot on a paper and kept in a closed chamber in such a way that the edge of filter paper is dipped into a developing solvent. The movement of the solvent takes place by capillary action, and the various substances are moved by developing solvent at a various speed due to its different partition coefficient or distribution coefficient in two different phases, that is stationary phase and mobile phase. When the solvent has moved this sample to a suitable height, stop the development and dry the paper, and spot are utilized by suitable reagent like neon hydrogen solution. So the movement of the substance relative to the solvent is expressed in terms of a RF value. So these are the requirements, that is chemicals in that phenylalanine, glycine, neon hydrogen reagent, methanol, acetic acid, and butanol, distilled water, ethanol, isopropyl, ammonia, etc. or other type of the solvents, nothing but chloroform, ethanol we can use as per the requirement. Then glass vase, because test tube, capillary tube, measuring cylinder, these are the different types of the main apparatus, glass vase we can use. The next apparatus, that is neon hydrogen spin apparatus. Then other is the Whatman filter paper in circular form, and pencil, then cotton we use. The next is procedure. So the first step is developing solvent was prepared, that is mobile phase we prepared and kept in a chamber, and chamber was kept for the saturation. We can use a filter paper wall for the saturation also. Then Whatman filter paper was taken and cut into appropriate circle or a radial shape, or we can also use ready-made circular paper. Then a hole was made at the center of the radial paper through the cotton tongue was inserted in such a way that it could dip into the solvent. We can insert the cotton tongue into that hole. Then from center, small concentric circle was marked, and by keeping the appropriate distance on this circle spot of a pure sample, solution and mixture of amino acid were applied with the intermittent drying. Then now this paper took horizontally on a petri dish so that the cotton tongue or the strip will dip into the solvent. Cover this paper with the other petri plate and allow the chromatograph to run. After achieving the sufficient solvent front, paper was removed and marked the solvent front position immediately with pencil. Then chromatograph was dried and then sprayed in anhydride solution and again dry it and gives colored spot of amino acid. And finally calculate the RF value of each amino acid by measuring the distance traveled by them from the line of origin, and ideally distance should measure from the line of origin to the center of the mid of appeared spot and the spots for the mixture. So this is the diagrammatic representation. We can easily understood how to giving the spot and how to use the wick and how to insert in it. So the direction of flow of this solvent is here due to this wick. We can also use the wick, or we can also cut a paper into such a way that it will form a tongue and we can dip into the solvent. So the sample spot, we can make a concentric circle and give the spot, and these are the solvent front occur after the completion of development, and this is the way of the capillary action takes place due to the wick or the paper tongue. Then these are the observation, that is you should write down whatever the mobile phase, and you can also write down the parameter for this RF value of glycine standard, phenylalanine standard, and the mixture of glycine and the mixture containing glycine and phenylalanine in a centimeter for the calculation of RF value and comparison of RF value. So distance traveled by the solute from the line of origin and distance traveled by the solvent from the line of origin in a centimeter. Calculation from formula for the RF value is nothing but distance traveled by the solute from the line of origin divided by the distance traveled by the solvent from the line of origin. Then these are the observations related to the glycine, that is RF value calculating. You can write down here phenylalanine for phenylalanine, for mixture you can write, and we can put the result in this format, that is a mobile phase we use, whatever the mobile phase we use right here. Then write down the respective mobile phase standard value of RF value for glycine, phenylalanine, and mixture glycine and phenylalanine as like this. These are the references we have used. So thank you all of you. I think you all understand this procedure. Thank you once again.